Extrachromosomal DNA, or ecDNA, are fragments of DNA that exist off of the chromosomes but inside the nucleus of a cell. These fragments of DNA have been linked to cancer, specifically aggressive forms of cancer that have been resistant to current cancer treatments. Being able to accurately identify ecDNA from the genome can help guide cancer treatments towards finding cures for these types of cancers. One characteristic of ecDNA that can help us identify it is that it is amplified. It being amplified means that the exact same string of base pairs appears at a very high frequency, much more than the rest of the genome. We would be able to easily identify ecDNA this way if it weren't for HSRs. HSRs are homogeneously staining regions, which are amplified strings of DNA on the chromosome. However, there is one major difference between ecDNA and HSRs that can tell them apart, and that has to do with genomic interactions. The key difference in interactions between ecDNA and HSR are that ecDNA, with their ability to float around the nucleus of the cell and interact with chromosomes freely, is that their interactions should be more uniformly distributed throughout the whole genome. With HSRs being on the chromosome, their interactions are more confined to that specific chromosome, with some limited interactions to genomic regions of other chromosomes. We can model genomic interactions through graphs, with each genomic region being represented as a node and each interaction being represented as an edge. This is known as high C data, or the adjacency matrix going along with the graph is a high C matrix. Previous work in identifying ecDNA has not considered graph structure or used any sort of graph networks to try to figure this out. However, there have been graph convolutional networks used on high C data in other research that have been able to accurately identify which genomic regions are active or inactive. Our problem, and what we want to solve, is we want to apply that work with GCNs, so graph convolutional networks or graph stage networks, and hopefully graph transformers, to the ecDNA problem, and use those to identify ecDNA through single-cell high C matrices. Graph transformers have performed particularly well on graph classification tasks in the past and in prior works. We hypothesize that the use of graph neural networks will be able to effectively classify which genomic regions contain ecDNA. Thank you.